Cucurbitacin B exerts anti-aging effects in yeast by regulating autophagy and oxidative stress. By Yuki Kotakeyama, Jingli Yanjun Panakira Matsuura Yoshikazu Ohaya, Minoru Yoshida, Lan Xiang, and Jian Huichi. Cucurbitacin is a bitter-tasting principle that can be isolated from members of the family Cucurbitaceae, such as cucumber, Cucumis sativus, and melon, Cucumis melo L. As a member of the Cucurbitaceae family of plants, cucumbers contain high levels of bitter-tasting nutrients known as cucurbitacin. Cucurbitacin is a class of biochemical compounds that some plants, notably members of the pumpkin and gourd family, Cucurbitaceae, produce and which function as a defense against herbivores. When cucumbers, muskmelons, and watermelons grow in the wild, their fruit contains bitter cucurbitacin molecules, a family of highly oxygenated tetracyclic triterpenes that deter pests from foraging. The budding yeast Saccharomyces cerevisiae has been used as a model organism for the basic mechanism of aging, which provides useful assay systems for measuring both replicative and chronological lifespans. In the course of our screening program for substances that extend replicative lifespan, Cucurbitacin B, Cub, was found as a hit compound from a compound library, which contains cerebrosides, phenols, sesquiterpenoid, triterpenoids, and sterols isolated from natural products by our research group. Importantly, it prolonged not only the replicative lifespan but also the chronological lifespan in yeast. Cub increased ATG32 gene expression, suggesting that Cub induces autophagy. Indeed, the GFP signal generated from the cleavage of GFP-ATG8, which is a signature of autophagy, was increased upon Cub treatment. On the other hand, Cub failed to increase the chronological lifespans when either ATG2 or ATG32, essential autophagy genes, was deleted, indicating that the lifespan extension by Cub depends on autophagy induction. Furthermore, Cub significantly increased superoxide dismutase, SOD, activity and the survival rate of yeast under oxidative stress, while it decreased the amount of reactive oxygen species, ROS, and malondialdehyde, MDA, production, indicating that Cub has activity to antagonize oxidative stress. Additionally, Cub did not affect replicative lifespans of SOD1, SOD2, UTH1, and SKN7 mutants with the K6001 background, indicating that aging-related genes including SOD1, SOD2, UTH1, and SKN7 participate in the anti-aging effect of Cub. These results suggest that Cub exerts anti-aging activity by regulating autophagy, ROS, antioxidative ability, and aging-related genes. Finally, we discuss the possible intracellular targets of Cub based on the phenotypic comparison between the Cub and global gene deletion databases. Yeast has the replicative lifespan and chronological lifespan. The replicative lifespan measures the number of daughters of a single mother cell, which can asexually produce prior to senescence. The chronological lifespan is defined as the length of time a yeast cell can survive in the non-dividing G0 state 1. Both of them are regulated by many environmental and genetic factors too. In our study, we used K6001 yeast as an aging model, which is a mutant yeast strain derived from W303. A unique character of K6001, in which only mother cells can produce daughter cells in glucose medium, but not in galactose medium 3, can be used for measuring replicative lifespan. Under the guidance of K6001 replicative lifespan assay, we have isolated several compounds with anti-aging activity from natural products 4, 5. Autophagy is a highly conserved pathway in organisms from yeast to human, which involves degradation of damaged organelles and proteins and circulation of amino acids and other metabolites 6. It regulates the genomic integrity via suppression of cell division in yeast under starvation 7. Besides, decreased autophagy leads to downregulation of protostasis, which is one of the hallmarks for aging. Dysfunction or decreased expression of autophagy genes leads to shorter lifespan in yeast and fruit fly. Conversely, enhanced autophagy promotes the longevity in aging models and protects against aging and age-related disorders 8. Especially, the lifespan extending effects of rapamycin in C. elegans, fruit fly, and mice are abolished by knocking out or knocking down of ATG genes 9. In addition, caloric restriction and resveratrol, res, induced autophagy dependent on SIR2 function in yeast and C. elegans, and the deleted and silenced ATG genes avoid the anti-aging effect of caloric restriction, res, and overexpression of SIR210. Therefore, autophagy has a close link to aging. Oxidative stress is another cause of aging. Free radicals produced through aerobic metabolism accumulate over time and accelerate aging. 11. In yeast, the accumulation of ROS promotes replicative and chronological aging. The upregulation of antioxidant removes excess ROS and maintains redox balance, eventually delaying the progression of aging in yeast. 12. In our previous studies, we showed that antioxidative activity plays an important role in the anti-aging effects of cholesterol and parishin. 4. 5. Cub has been shown as a promising compound for anti-cancer treatment in recent years, and it also shows antioxidative and anti-inflammatory activities. 13. 14. The regulation of STAT3 and RAF/MEK/ERK signaling pathways by Cub is likely to be involved in its anti-tumor activity, such as induction of apoptosis in tumor cells. 15. Moreover, Cub was found to induce autophagy in cells. 16. In the present study, we identified Cub as a compound with anti-aging activity in yeast, resulting from our screening for anti-aging compounds from a compound library using yeast replicative and chronological lifespan assays. Here, we report a unique activity of Cub, which exerts anti-aging effects in yeast through regulating autophagy and oxidative stress. In China, Cub has been used to treat hepatitis for many years. 37. Cub also has potent anti-cancer, antioxidative, and anti-inflammatory activities. 13. 14. In the present study, we performed replicative lifespan assays to screen for anti-aging substances from compounds library and used chronological lifespan assay to confirm the activity. We identified Cub as a potential anti-aging compound. The chronological and replicative lifespan results shown in figures 1b and 1c imply that Cub exerts significant anti-aging effects in a dose-dependent manner. We previously applied the replicative lifespan assay to evaluate the anti-aging activities of other compounds. However, we found that this assay exhibits deficiencies that may affect the accuracy of its results. For example, random selection and subjective factors affect the quantification of daughter cells in the replicative lifespan assay. Therefore, to increase the accuracy of the bioassay results in the present study, we utilized the chronological lifespan assay to evaluate the anti-aging activity of Cub. Yeasts have replicative and chronological lifespans. Replicative aging in yeast resembles the aging of mitotic cells in higher organisms, and chronological aging is similar to the aging of non-dividing cells in higher eukaryotes. 38. The results of replicative and chronological lifespan assays suggest that Cub affects both the replicative and chronological lifespans of yeast. Autophagy levels decrease with age, and autophagy activation ameliorates age-related symptoms and has potential functions against aging. The results of gene expression, replicative lifespan, and chronological lifespan suggest that ATG2 and ATG32 genes have essential roles in the anti-aging activity of Cub. Besides, enhanced mitophagy contributes to the extension of longevity by Cub. We also investigated the effect of antioxidative activity on the anti-aging function of Cub. The changes in the survival rate and SOD, ROS, and MDA levels of yeast demonstrate that Cub exerts its anti-aging effect in yeast by regulating antioxidative activity. Longevity-related and aging-related genes have important roles in the regulation of aging and longevity. In this sense, the possible targets of Cub are not associated with cell growth or cell morphology. Nevertheless, additional efforts are needed to definitively identify the targets of Cub. 5 Conclusions. Overall, we found that Cub significantly prolongs the replicative and chronological lifespans of yeast in a dose-dependent manner. Cub exerts its anti-aging effect by regulating autophagy and antioxidative activity. Moreover, the target identification of CUB suggests that 44 proteins that are not associated with cell growth or morphology are potential targets of CUB. However, the specific target of CUB requires further study, such as bioassay on these 44 proteins. Furthermore, given that CUB is already used to treat hepatitis cases in China, it may be developed as an anti-aging drug.